Very much, Ron. It's a pleasure to be here. It's post-COVID, so I kind of appreciate traveling and not being on Zoom for some of these conferences. So my talk is about CMOS biochips and the history of it. When I was preparing this talk, I've been in this field for 24 years, I guess. It was when I was a graduate student. Some of you are graduate students, I guess. It was just starting. So when I was going through my notes and preparing this talk, I was walking down the memory lane, and some of these things hurt a little bit. Some of them were just more fun, but I tried to put all of them together, and I hope you'll see what has been going on in this field for a while. But what is CMOS biochips, or why do you use the word CMOS into it and all of them? So today, when you look at it, there are some products specifically in the biotech domain for sequencing, molecular analysis, that they have a kind of a semiconductor chip. These are not implants. These are not things like that. And essentially, we call them CMOS biochip. Why do we use the word CMOS? This is a historical reason for it, because the word biochip, as you will see, has been used for a lot of things. So at one point around 2000, I think about 4 or 5 people said, okay, we have to somehow tell everybody that this is a semiconductor device with electronics, with metallization layer, and everything is there, and it's built in a standard foundry. So they, boom, put the word CMOS in front of it. And that time, the words like CMOS RF were common, because everybody says, okay, they're going to be CMOS. So the history of the CMOS part of it is part of that. But the application, you will see that, obviously the biochips, there are many different types, but this is a CMOS type of it. So what is the motivation? I was trained as an electrical engineering. I live and pray to the gods of EE, I guess. And we all know that in our field, at least in the circuit design part of it, we have a lot of stuff that are pretty robust. We have best in class in manufacturing for the wafer level processes that we have. They're available, they're good, they're scalable. Some of the process nodes, specifically the ones which are used for sensor nodes, are cheaper right now. And we also have the best kind of sensors that in your phone and a lot of things you use. So we have a lot of things which are kind of sensorish, and we know how to build a lot. And there's always been a motivation for people to see that, can we apply what we already have for other areas? And that's the very important topic. We don't want to reinvent the whole manufacturing for something new. And I think the motivation for this field was kind of use whatever you have and see that if you can find an application that has an impact, and also market that would be relevant in this area. And I think diagnostic, pharma research, and some agricultural food application, because of the volume, are interesting. So I want to differentiate over here. These areas do not go to implants, for example. They don't have the volume. These are very special areas that we have. So that's kind of in the field, that's how we differentiate. This might be a little bit of a nuance, but that's where the biochips are. So, and when you talk about biosensors, I have to talk about our perspective toward, this area of biosensing and application. I generally use this slide because there's a lot in it, but I just want to show you some things which are relevant. First of all, there's a lot of targets that we can detect. People throw all these targets on a biosensor and, they can detect it. But one of the things that you always have to remember as far as the applications, if you just look at the concentration of things, and if you assume in 1 milliliter how much stuff I have in water, 1 milliliter of water, you have this much 10 to the power of 22 molecules of water. When you go to glucose, it's 10 to the power of 18. You might not have a feeling about what 10 to the power of 18 is, but you can taste it. 10 to the power of 18 is a lot. But let's look at some other applications. The concentrations of orders of magnitude or less. So what is my point over there? So the application, or the way that we do the sensing, as far as just concentration, is different. So when you see a glucose sensor that you can do, probably not everything is going to be used with the same way. Sometimes have to have different techniques, and deal with it. And I'll briefly touch on top of it. The other type is there's a lot of strains. There are different types that you have to detect. We're going to touch on that when we deal with a little bit of the specificity issue. But the part for an engineer or people who are in applied domain like me is relevant, is generally the idea of, does it make sense to use this big hammer of a semiconductor for that? It might sound not very academic, but it's very realistic, that you have to look at, as far as the volume, as far as the prices and everything, is something that worth doing. Because if it doesn't have the volume, or it doesn't have commercially, there's not an impact, sometimes your research would be kind of one-off, and you have to make it, and doesn't make sense to build a chip for it. Just basically the economics doesn't make sense. And there are applications, obviously, that when I make it red, that or make it, bold green, that there's a lot of application of it. One interesting one was COVID that we all knew, but the number is probably, I checked before my travel, this number is less than this right now. So probably in the US is getting to 300 million for a PCR test, but these things go up and down by the nature of this. So this is the context. This is what we want to go for, and let's just look at, what I want to talk about. First detour I want to make, I want to talk about a bunch of figures of merit that I care about, and I might throw it in the middle of the talk. I just want to make sure that we're all on the same page. First of all, on the biosensing, sort of domain, when you look at it, there are many different types, enzymatic, there are kinetic-based, but the classical one that you basically capture something, there is an energy change between the target and the probe that you design, and basically that energy difference by diffusion or whatever process that they get near it, you create a complex that you want to detect. In the case of all this DNA sensors and everything, independent of the application, sequencing and everything, the same concept applies. So you look at the energetic of some molecular recognition to do that. But the implication of this on a sensor is more than just that. In most cases of biosensing and in the CMOS biochip that we'll talk, you will dealing with the same problems that they have. One of the first ones that you deal with, and this is a classical when you teach, that if you have a non-specific target, you will have the same complex or similar complex detectable can be formed, but with less probability. And this is based on the energetic, and you might saturate the surface or give you a background. But this is one of the things that, in the case of biosensing, pushed us toward using very specific probes or specific probe structure, be it nucleic acid, DNA or PNA, and in some cases why people are using antibody and everything. And the reason is this, and this chart is trying to explain this. If you look at the number of targets that you want to detect, and also you have backgrounds that they might bind, depending on the energy difference of that, there's going to be a curve that shows where you get the signal to background of what. If you plot that curve based on the energy difference as a function of KT, you will see that biosensors are not very good sensors, just based on biology. You say biology very specific, well, to some degree. If you look at DNA, if you want to measure a single mismatch in that, probably if you're lucky, you get 3 KT. 3 KT means that your signal to background of, 2, you can detect. If you look at the aptamers a little bit better, proteins are all better. So when you go to the application that the signal to background, that means the similar molecules are high concentration, these things fell off the wagon. So a lot of the assay design and the methods that you deal with is over there. And when we go to the CMOS biochip, sometimes we have to deal with these in the functionality of sensors to be able to accommodate a good sense. For example, the reason that people use amplification, signal amplification, they do titration, and all of them is not because they're just amplifying signal. Sometimes they're dealing with this issue. So just remember this. The other issue that is well known and has been one of the biggest problem in some of the platforms like microarrays and everything that we discuss about it, biosensors, especially the ones which sort of rely on capturing and detect, they're defined by Langmuir isotherm, and basically they saturate. That means that if your concentration of the molecule goes up, depending on the equilibrium constant that you have, at one point, this is going to be pretty flat. You're going to saturate the surface. So what does it matter? If you look at your signal processing and noise kind of analysis 101, when you have a signal that flattens out, and if you have a little bit added noise from your instrument or anything over there, automatically your signal to noise, we're going to have a minimum detection level, which is a point that your signal is small enough compared to your sensor. And when you saturate, it becomes flat, and you cannot distinguish if the concentration is higher or lower. So you get a pretty limited dynamic range. This dynamic range is very low. In best case, it's 2.5 order of magnitude that I've seen in some antibody array sandwich assays, but generally it's around, probably 1.5. So you cannot quantitatively look at this. Okay, so this is pretty depressing. So this is one part that I wish we could talk about this presentation. This is not my job, but one of the things that I always remember, there's the creativity of people in this field sometimes to solve the sensing problem has been immense. One of my, for example, when they want to figure out the issue of selectivity, that means remember that KT pestering thing that I mentioned over there. They don't rely on one reaction to get the positive response. They essentially create a biological and. So this reaction should happen, second reaction should happen, third reaction should happen. All of them are specific, and the aggregate of that will give you the signal that you want. There's a famous paper, there's the work that came out of Chiron. I think it's a textbook brilliance, pre-PCR area, pre all of them, which was done in 90s. They wanted to detect the HIV at that time without all of them. So they created something called branch DNA. The fancy way of, basically what they do, they capture the molecule. If the molecule is captured, they have a probe that on a different location of the molecule, in this case, the DNA captures, and then they build a Christmas tree of fluorescent molecules on it. And they were able to detect probably 40 or 50 molecules in one reaction at that time without any amplification. And it was pretty specific, 3 reactions of DNA, 3 KTs independently. That gives you very, very specific if you design them correctly. So there were ways of doing it. When it goes to the quantification range, remember that long-term isotherm, people use, classical, when they do the, enzyme detection of antibodies and antigen detection, they do the titration series. When you go to the nucleic acid amplification, they do the reverse. They don't reduce the concentration. They increase it by PCR, and they start looking at it in every cycle. So in that case, you are having something which is a limited dynamic range, but you're using it. So these methods, in conjunction with the platforms that we're going to talk about from now, is going to make sense. So when we talk about the sensor, when I talk about signals, noise, and everything, when we talk about it, just remember that there's no way a single chip or a sensor will do that, and there has to be some of these methods that you have to add on it. These methods, generally in the biotech, probably 90s or 2000 when you came in, they're all patented, and a lot of people were trying to get around it. But right now, all of them are available, so you can actually look at it. But if you're in this field as a student, take a look at these methods. Some of them are pretty inspiring, and it shows true ingenuity of people. All right, so that was the start of it, but let's get to the, sort of what I want to talk about, my history of what happened. I basically decided to divide the history between the ancient times and what we're going to talk about it, and I just arbitrarily put it at 2000. Well, that's the time that before that I really don't have a first-hand experience. I was just, not a graduate student, but after that, I remember. But if you look at this time, there's a lot of things that we can talk about it when it comes to biosensor, the first commercial biosensor. Clark made the oxygen electrodes, Sanger, somewhere around here, they invented the chain termination method for DNA sequencing. Polymerase chain reaction, in 83, they started to do that. So there was a lot of component that happened. But what really happened around 2000 was the first big biotech boom, which fueled actually the CMOS Biochips that we will talk about it. And this was, the same time that Human Genome Project was finished, and we're going to start from there to see what happened. The first thing that you want to know when you get to this area, you have to know a little bit about what was the DNA microarrays and what happens. Today is less relevant, but historically is I would say, the grandfather of a lot of things that we use today. So at this time, I would say, if you look at the history of it, probably the paper or the work that you want to talk about it is the first time that they made a high density array of DNA to capture, sort of the cDNA, the mRNA in this case, on a cDNA array to create it. The relevance of that, and this work was done in Stanford, these guys. The first guy is in prison right now, I'll tell you. No, he is. He did something bad during COVID, and, they went over there. The last guy, Patrick Brown, have you had the Impossible Burger? How many of you have Impossible? That's his company. He just rage quit Stanford and started that. So you can look at it, Patrick. The other two are not that, well, they're big shots, but they're not that interesting. But these guys were postdocs, and 2 faculty, Ron Davis and Pat Brown, were faculty over there. So 2 postdocs, Mark Shenna, and they put them through a lot of abuse, and essentially they create an arrayer. They put pins that come and essentially place a bunch of cDNA surface of glass, and they showed that they can create a high density array to detect with a fluorescent detection, so the mRNA expression in a cell and everything. This was very interesting because this was the first time that they did it in high density, right? So before that, there were obviously these methods were not new, but they did it high density, and it was the first time people could say, okay, now I can look at a cell and see what exactly happens at different conditions and everything. You go. So this started this whole array mania that everybody wants to create a large array. The first company that actually commercialized it was Affymetrix. They were very relevant when I was a graduate student. People would pray to their altar, and
I can't seem to log in.
To conference
Yeah,
desk?
even for Wi-Fi.
Can I see your Wi-Fi?
hello?
Yes.
Is this? I don't know. I think we were interfering with the, I'm a speaker somewhere. Yes. Okay. Oh, go ahead. Caroline, don't do that. Yeah. All right. So, basically, so Affymetrix, what it does, they want to make an array and commercialize it and everything. So one of the challenge was, if I want to put, thousands of different molecules on the surface, how are we going to do it? I'm going to go one by one, pick it up. Yes, for a graduate student that you put them through abuse, they can do it, but commercially, that's not doable. So they came up, and there were a lot of, brilliant chemists. They came up with this method that is shown here on the right. They essentially said, okay, DNA has 4 bases, and we want to make a DNA. Why don't we synthesize this thing chemically, and use some optical method to protect and everything to create a DNA sequence? Do it. So they came up with the chemistry. Now they have to make an array. They started using a lithographic method. So they had masks for each of them, and they sequentially would add the molecules, deprotect, add the chemistry, go 1 base up, and they would actually do. So if you want to make a 20 base pair, I guess you needed 80 masks to do it. It was very exciting because as far as scale, you could make a lot of them in parallel. There was a lot of issues with it. The quality of the sequences were crappy because you had 1% error at the beginning and all of them. But they created high-density arrays. High-density arrays, and they went and did a lot of patent. They even patented the distance. So Affymetrix, you could not make an array with less than 100 micron. That was one of the main things that I got. And they commercialized these things, and for detection, they use fluorescent detection. So they would stain the molecules. This was the biggest thing, and everybody wants to make an array, the high-density array. In university, or some smaller setting, people went extreme to using robotics to do the print. Especially initially, they had contact printing, so they would go touch it. And by the way, I forgot to mention, do you see the donut shapes on that one? That's the tip of the thing that's hitting on the surface. When you see donut shape, that's printed by touch. When you don't see donut shape, those are generally droplets or some other. Anyways. But so they make the arrayers, and one of the issues with the array is what you had to load. So you had to pick what you want over there. And imagine when you had 100,000, you have to do 100,000 pickup and everything to do it. So at that time, the ancients were the other options. So a company called Protogen, which didn't survive. They went back to Agilent after they ran out of money. But the technology was generally do the inkjet printing. So you do the ACGT, you have 4 colors and do it, and you deprotect and everything, and they created the arrays over it. They were going full speed. Now, out of the blue, this is a company that is forgotten, but I want them to survive because there are some lessons in there. Actually, the first chip that anybody built in the bio, it was by a company called Affymetrix. The idea came out of one scientist in Caltech, and he wanted to use electrochemical methods to synthesize the same thing that Affymetrix was doing. So they created an electrode array. It was a platinum array. I'm 90% sure, I couldn't get the confirmation before this talk. This was built in the same foundry that the Coder 64 was built. This was a MOS technology in Pittsburgh. But the idea was essentially you add the solution. If you apply voltage, you're going to get the current and you're going to induce some pH level over there. The chemistry was activated by pH. So you have this electrode array that you would address a different location. You would put the thing and you would start synthesizing it. The company went out of business. It was just too expensive, too erroneous. But it's an interesting idea. And I think in San Diego, some of the faculty are reviving this project, which is interesting. The difference between this and Affymetrix was this technology, you essentially could design your own array. You didn't need to have a mask set built for you. You could program it. So they were commercializing this as a customer, which was interesting. Now, another technology came out of it, which is a little bit complicated, but it's worth mentioning because it's the kind of the father, not the grandfather of a lot of things that we deal with out of it. And it's the random array that Illumina came up. So Illumina was founded when a guy that left actually Affymetrix started Illumina to do this, an array. The idea was this. I don't want to synthesize on the surface. So I get these small beads, silicon beads or silica beads. I will put the DNA that I want on it. So I would functionalize it and with whatever I wanted to it. On a surface, I make some holes and throw this on it, and they're going to go in this location, and I create an array. When the first you look at it and say, whoa, wait a minute. You just throw it on the array. You don't know which one goes where. It's a random array. But the idea that they had was essentially on the probe that I put over there, I can put a barcode or a known sequence that I can add, complementary sequence at the end of manufacturing to basically find out which probe goes where. It might sound crazy, but manufacturing was one of the best things. Why? You could design any probe that you wanted. So you were not limited by that synthesis. You can do a batch of one type. You can make this much of it whenever you want to use it. And this barcoding and identification is not what the customer do. You do it in the foundry, and you can QC the array too. Initially, people said it's crazy, but guess what? This is the one that was the winner out of all of them. So it sounds complicated, but it solved a bunch of problems. And at the end of the arrays, people who had performance and everything would win. We'll touch base on that and the conclusion of it. Now, during this time, there was another issue that was happening. I mentioned all of these are fluorescence-based detection that they use with scanners and everything. But the fluorophores at that time, a lot of them were under patent, cyanine dyes, and all of them that they use Site 3, Site 5, well, the ABI patents, and they would charge you an arm and leg. So the royalties were pretty high. So there was a parallel group that actually I was more on that side of the detection. They wanted to use other methods of detection. And sometimes they called them label-free. That didn't mean they didn't have a reporter necessarily. They wanted to stay away from fluorescent label and have it. So people have, a bunch of electrochemical methods were developed. I think clinical microsensor right now became GenMark, redox-based, ferrocene, methylene blue, and a bunch of them were at that point. Crazy ideas of, SPR on an array. I don't know, however you can do these high-density arrays. So people were doing this as well during that. But at the end of the day, the conclusion was this. Well, when we're doing high-density arrays, we're looking at an organism and everything we do. There's no customization for the probes. So the fixed arrays end up becoming random microarrays. So around 2005, I think Illumina passed Affymetrix on the array, and it just went to the sunset, and I think bigger arrays right now are fixed arrays that already they're built, high quality, and use Agilent has some of them and everything. But people who wanted to do custom arrays, they ended up doing the printing. Because at that time, you had something special that you wanted to design, or you were 100 or 200. So still the university groups and a lot of people even who work on the protein side of things that you cannot synthesize. Well, there were some effort like Health Cell, they tried to synthesize peptides and make arrays. They didn't that much work out, but essentially they went to that type of array, and patents for fluorescent labels, they all expired. So the Alexa dyes came in and everything. So people basically, that was the conclusion around 2005 and everything. But one thing which was bad, and I think was a killer on arrays, was people did the gene expression, did which gene is upregulating and everything, but they needed a more quantitative, analysis. And this would not cut it. So at that time, they labeled microarrays, and it's still there, semi-quantitative, or whatever that means. And people moved to qPCR methods for a smaller number of genes, and then sequencing came in, and the dynamics changed. But that was the history of it. But remember, the arrays were there. Okay, so during this time, you see electrical engineers or people on the engineering on the other side are smart. They're looking at all these bio stuff that are happening, and they start looking at it. Okay, what's my cut? What am I gonna do? And I think the whole idea of CMOS Biochips started over there. You see, somebody who knows memory or image sensor deals with an array, right? So you look at these arrays and say, can I somehow fit these into this? I'm simplifying it, but a lot of the efforts were over there. I think, the first CMOS biochip that was designed and fairly published was done in 2004. And I think this is from Infineon and Roland. Where's Roland? Roland's over there. So you can ask him and give him a hard time about this work that we have. So they built this one, and it's an array. Yeah, you see an array, and it basically uses alkaline phosphatase and for detection. So it's a DNA-based, and basically you have your interdigital electrode, you apply voltage when it comes in, substrate, you get a current, and basically you actually detection. It was pretty, at that time, that was a big deal. Wow, it's the first time, and you had the switches, you can see the blocks of it, and this was published in our ISSC, which is for us, for the circuit designers, the biggest one, and it just off to the races. After this, it started. this was a lot of people at that time, and still do, they think electronics, they think everything is electrical. You cannot do anything in optics. But I think another work that came out of it at that time was another group here in the US where I was there with I was part of it. They started doing this, can we do optical as well? So they make an array, which was a glorified image sensor. This was a 3 transistor even pixel that they want to have. And they wanted to do the pyrosequencing, which is one of the methods that you can do sequence by synthesis to basically do the detection of the light for an array, but it's a microwell pace. And they did it a year after that. So when I looked at it, it was like, okay, they're getting there. We have the basic elements, and after that, things start. So those sessions in those years, which was between, I think, 2004 and 2010, there was a lot of ideas. Most of them, including the work I did, and all of them were half-cooked. You didn't know. But people were showing that it's possible. It's possible. They did the, time result fluorescence, with electrochemical, voltammetry, all those methods that you could do. And everybody has a different angle. But, if you look at it, the yellows, kind of rectangulars that I put over there, they all had an array. You see the microarrays, the arrays were over there. Implicitly, we're telling you, if you're using a chip for this volume, I need to have enough detectors. Just one detector for a chip, it just doesn't make sense. So they were understanding that if you have a problem, has to be big enough, worth it, an array to be able to do. And other thing came out of it. There's a work from Milgro that I'm going to talk about. There was a pH base. Initially, we were saying, what the hell is this? Why do you want to detect pH in an array? This is actually an important work that was done over here. And then I was in Caltech. Some graduate students, when they said, oh, I know RF, I want to make a magnetic. I said, why? Why do you want to magnetic? He said, well, I just want to do. So they put a magnetic bead to be able to detect biosensing with a magnetic bead. Well, I don't recommend it. The bead in a presentation, a magnetic bead as a label looks very small, but in reality, just what, 10 million Dalton or something. It's like connecting a basketball to a fly, and you expect the fly to move around. You cannot. But still, we have a magnetic biosensor in this case with the magnetic label. And then there was a work, I was working probably fluorescence. The fluorescence, you have to create an emission filter over there. But around, I would say this time, 2004 and 5, there was a lot of work has been done. And I think at that point, if you review those articles, you know where you stand as far as the CMOS part of it when it compares to modes of detection. This is standard CMOS a little bit. And this slide is a little bit, crowded. I apologize, but I think it's worth looking at it, and later you can look at it as a reference. In that publication, all the references for each work are over there. So on the optical side, we had the fluorescence and the bioluminescence, luciferase based, or any of those things that you can look. You can use the photodiodes that you have and everything that you can do on the image sensor they have. There are different sort of front ends that people design. And one of the good things about all these biosensors is the rate that you have to do the sensing is not very fast. So generally, sort of a 50, 20 samples per second is the overkill in most cases that you're looking at something at equilibrium. You can do a measurement every few seconds. So the Nyquist rate of the signals are not that very high. But all of these methods, they may or may not need a processing step, besides the molecular biology and everything, that you have to do on the chip to make that sensor function. Sensor, I'm talking about the physical sensor of it, not the bio part of it, the sensor part of it. For example, if you want to do a fluorescence-based detection and you shine in an excitation light, you have to block the excitation light and let the emission to go. You need a good filter, optical filter, which is wavelength selective. CMOS processes do not give you that. So you have to do post-CMOS, you have to do another layer on the wafer or whatever that you do. Generally, these are the things that limit the manufacturing and sometimes the cost of these things. Because the chip, we know how much it is. This one comes in and it's a custom. You go to foundry and you want to do custom. They look at how many commas you have in your bank account. It's just sometimes not economically doable. But bioluminescence you don't need. There's no background, right? Well, bioluminescence is not as widely used, but if you want to use a bioluminescence assay, you can get the cheapest, it's basically camera. Now, do you want to do focal plane array or you want to put the molecules on top of it? It's up to you. But these were the kind of ideas, and this was fine. This was an optical filter. When you go to the electrochemical, or just fancy word of it, electroanalytical, you have voltammetry maze, you have charge-based sensing, impedance spectroscopy, And all the methods, and this was a pretty rich area because obviously in electronics you can do it. But, I think still the electrode manufacturing and everything is an issue, and today is still an issue. Because there were not that much commercial success in this area, at least in this field. We'll talk about it a little bit, but this is an issue. And when you go to the magnetic, I just put it because I needed 3 columns. I don't think there's that much in it, but, just had to put it over there. But generally, that's the 3 areas. And if you look at it, I think the challenges are essentially this. Again, not the bio part. So if you look at the fluorescence-based one, yeah, optical is an issue. This one, the electrodes, we'll talk about it. And some of these things are crazy materials that, I don't understand. I don't understand any of the papers, but probably they're interesting. But you have to make them. The other thing that you have to remember, beside the background, the bio that I talk about, you have to also be aware that these sensors, when you put it in the context of the bio application, they also have an additional background. And background, what I mean is the fixed signal that you get and the fluctuation of that fixed signal to temperature variation during the time, and sometimes that becomes your limiting. So in the fluorescence, obviously, is the background light. If you just imagine the fluorescent excitation, 6 orders of magnitude, at least more than your signal that you're detecting. So a little bit of that thing leaking, it just overwhelms everything that you have. And when you go to the electrodes, if you do cyclic voltammetry and everything, if you have worked in that area, first of all, I would respect you. You have gone through a lot of pain and suffering. A few angels have died for you when you do work for them. But that electrode has a personality. Just drifts and go over there and all. By the way, everywhere it's an issue. And when you put into cyclic voltammetry and you go over there, suddenly everything disappears. So yeah, that's one of the issues. And of course, we can talk about it if you ever have questions what is coming about this area. There might be some solution about that we have. But that was the idea. So what happened during that time? You remember microarrays were going down, industry was going down. Remember, this was after the dot-com bust, so everybody was going down. Everything was going over there. But what happens in this area, this next generation sequencing come up? It's relevant because I'm going to tie that back to the biochips and everything over there. So at that time, remember, we had arrays. Microarrays were going out of business. Not out of business, out of the hottest thing, out there. So what the other thing which was very obviously relevant, and it was over there, was DNA sequencing. At that time, around 2000, I would say 1, 2 that I remember, which was the Human Genome Project. Everything was about the Sanger sequencer. Kudos to him over here that, basically, and the dominant method of sequencing was that, and the dominant instrument for that was the ABI, Applied Biosystem Sequencer System, which most of the Human Genome Project was done by that. The idea was, and this is relevant because, pay attention to it. So let's say I want to sequence something which is very large, 3 billion base pairs. What you do in a sequencing method, and Sanger is no exception, you go enzymatically or some other method, with shear force and everything. You tear it apart at different small parts. And if you can sequence these small parts, look at what it is. If you do enough number of the original, you can put the puzzle together because they might have overlaps as far as the sequence, and you can assemble. So in Human Genome Project, they did that for 3 billion. But the throughput of their instruments, best case, this is the best case. This was not what they started. But they could do fragments of 1,000 base pairs using capillary electrophoresis. And they could use probably 96 a day. So every day you get 100 kilo base pairs, you could do. So if you do the calculation, it's going to take years, which took years to do the whole thing. But this was interesting. But remember those random arrays, the weird ones? There was a company came out, which was the name of the company is 454. I don't know how many remembered 454. The name of the company was 454 because the company came out of another company called Couragen, and they had a lot of internal projects. This project number was 454. So 454 was the number of projects. So 454, you have to give them credit. They created the first next generation DNA sequencer. They had the array, remember the Illumina array, that you didn't know where things are going. If you can sequence in it well, sort of using optics, in NGS or next generation sequencing, you don't care which one goes over where because you already have randomized your cutting. So they look at that, your problem is random. I'm going to assemble it in software anyway. So they essentially created this random array. And this was a crazy idea. According to the people on that team, the 2 miracles should have happened for this system to work. And I guess 2 miracles happened. One of them was, making the array work. And these, beads that they capture, and they did the emulsion PCR, doesn't matter. But they captured one sequence, hopefully, but it's statistical. So they had to dilute the solution that at least they get one over there. So the array was, 2/3 of the array was empty. So they had to capture it, and they add the solution, and they do an iterative process. They look at chemically and look at the optical that comes out of it, the photons that comes out of it. And this method was a sequence by synthesis combined with the array that they made the sequencing. But the arrays were over there, and they were about 44 micron. These small things that you see are not the beads. These are where the enzyme delivery system is. So they immobilize the enzyme on small beads to be able to localize the enzyme, which is crazy. But they did, and they could do this thing in a matter of, much faster. They could do it in 2 weeks. So, and this work, obviously, it started the whole NGS area. But the person who came up with the sequence by synthesis, there's one person called E.D. Hyman. I want to mention that in all the talks I mentioned. This is a guy who was a graduate student who left out of rage, out of his group. And he had only one publication in his career. In 1988, came up with this sequence by synthesis, and he disappeared. I tracked him down 20-something years after that, and he's working in Louisiana somewhere. He's doing antibody synthesis, and he never did anything else. And if you call him up, he's very salty because he didn't make anything out of it, but he came out of it. So this paper is it's something which is probably, if you look at it, it's 20 years ahead of its time. But yeah, a brilliant guy who I think, fell off the wagon or something over there. But these technologies were made. So when this array came out of it, obviously Illumina, remember they had array? They just jumped on it, and they got Solexa, which is a British company over there that they had the reversible terminator. They got the reversible terminator, and they decided smartly that we're not going to use the beads. We're going to put it on a surface, and it doesn't have to be exactly location of things that are created on the array at the same time. We're going to image it with very high resolution and identify where the spots are. So the first Illumina sequencers will build based on Solexa. And again, like always, Illumina, I think these are in their history was there. They had brilliant engineer, extremely good chemist, and they created the systems of sequencing that you see over there, and they could go to 100 gigabases per second. So you could sequence the genome in 2 days. And that's what we hear about it, but the history comes on, and 454 was sold to Roche. They couldn't, push it forward, so just fizzled away. So Illumina stayed, and these arrays were pretty interesting. And this thing was happening out of nowhere. Well, I know to give you a disclaimer, I get some royalties out of this for the work that we have, but in 2011, there was a small company, which was the founder of it was Rothberg, which was the founder of 454. They came and they introduced this, which was the first, I think, CMOS Biochip for next generation DNA sequence. They created an array, and basically the chemistry that they run was an electrochemical base. But there are a couple of interesting things about this one. First of all, the arrays came out of 454 because they knew about it, and the reaction they were running was sequence by synthesis that they had. Remember that Milgro paper that I mentioned? That pH sensing that I said, if you look at it, is identical. And I know some of the people over there joined to build their first chip, the 314 of it. So if you look at that, it's not an ISFET. I don't like to call this an ISFET. It's a floating gate. It's a matter of the difference that we use of the language. And basically they had it, and they generally measured the perturbance. I'll talk about it in a second. But the array essentially was a 1.2 million pixel. So it was not billion. It was not much larger. And they created, everybody got excited because it was a real application up and coming, and it was out of that microarray domain. And the sensor looked simple, and you didn't have to do that much surface chemistry and everything. But when you look under the hood, there was a lot of details. So although it looks simple, first of all, when you look at the chemistry of it, and you look at it on this floating gate, although they generally say that it's a pH sensing and everything, it doesn't do in equilibrium, you don't have any signal. So it's not changing anything in double layer like that. sort of equation that we write and everything. Essentially, you're detecting diffusion potential. It's capacitively coupled to the surface. So when the reaction happens, you create pyrophosphate and you create one hydrogen atom out of equilibrium, and they have to set. Because the buffer, if you have a buffer system and everything, you have to settle. So you have protons, like you create two carriers in a semiconductor. Well, something happens to them as they diffuse, and they can recombine, obviously, in all of them. In this case, you have protons that move fast and pyrophosphate moving slow. The showing of this reaction working on the chips and everything we've been done, but they could never make before this book, they could never make the protons detectable because they used a buffered solution. When you have a buffer solution, the molecules are controlling the pH. So if you create, they would recombine like that. So you would see the negative effect of pyrophosphate. If you look at the old paper in this area, which they smartly do not cite, you see the negative charge in all of them. But in this case, they debuffer it, and they created the positive charge that you can see out of proton. When they debuffer it, just getting into that, what happened to it is the read length was initially very low because they messed up the enzymes and everything, because all of them have to be over. So initially it was a turbulent start, but it was getting, and everybody was working on the chemistry, but it was huge excitement. Everybody was happy this thing will come out. But it started a debate and said, oh, okay, so this is my personal sequencer. Is it $25,000 versus $150,000 that I have to buy from Illumina? So everybody bought the instrument. But when you bought an instrument, you have to think about the performance and everything. That was the thing that they started asking question, should I actually do a sequencer? Should I actually have a sequencer, or basically, go buy something from Illumina? Which one is better? Also, the other question that came out of it was, okay, if this array gets bigger, if it becomes bigger than the size of a radical of a manufacturing in a way, what are you going to deal with? And when you have, sort of 100 megapixels, what rate I have to get the information out of it? Because this was a kinetic assay. Remember, it was nothing in equilibrium you had to do. So the I/O becomes an issue. The size of the well, if you reduce it, the 1/f noise of that floating gate would go to the roof. You wouldn't be able to detect, that much stuff into it. So, but a lot of people just conveniently ignore this. And they said, okay, we're going to go and beat Illumina and everything. But at the end of the day, well, the dominant method end up being the methods for DNA sequencing, the one that people to first order do not care about the size of the instrument, especially when you want to do whole genome or some big project. You just load it up, and somewhere in any university in a basement, there are two technicians plus one old postdoc that can run those things. So you send it and you get it over there. So, and that has been the case. But when people wanted to do something which is more targeted, and let's say I want to look at just a few genes, or I want to do pre-amplify something over there, you might actually use a different method that you wanted to use. And that was the time that maybe the semiconductor-based sequencing comes in. The whole Oxford nanopore and all the nanopore saga is also focusing on like that. So similar to microarray, remember, if you have fixed content, big iron, you don't need it. And the best analogy that I've heard, do you need a server farm, at your company? Not sometimes, no, you don't need it, something like that. You just need the right computation over there. But this decentralized diagnostic and applications were coming on, and if you want to have a sample to answer solution, and that's where it ended up. And this is around 2015. This is a few years before COVID and everything. And I remember that time people were worried. What's coming next? A lot of people put investment and everything in new technologies, and they could see that, now it's a little bit different because we all experience infectious disease. At that time, everybody was working about cancer. A lot of things were about that, application or funding was over there. There's not that much sense of urgency. Your lump over here in the next 24 hours is not going to, something not weird is going to happen. So it was a different dynamics. But, and what happened, I would say, after 2015, and still we're going through. So I think it's too close in the history that I can, sort of deal with it completely objectively. But people at that time started, okay, we have to have precision medicine. We have to look at things that we want to do, precisely with high precision, with high dynamic range, with accuracy, and essentially go from there. And this was a time that a few other systems came out of the world, and this was the kind of the PCR-based methods. They were coming in. People were talking about multiplex PCR. So what am I talking about? Is that when you look at sequencing or some of those applications that I just explained to you, you do not have any a priori information about what you're going to get. You're going to go and sequence and look at everything, put it together, and then look at the path. So when you're doing a sequencing, a general method, you look at all the nucleic acid which are available or all the targets over there, and then you can find out it. But when you're looking at application in diagnostic, by the nature of it, is hypothesis driven. When you're going over there, you're not looking at that person. Let's test everything in the universe. No, they say, okay, you have a sort of runny nose, probably is upper or lower respiratory, something is wrong. And, probably, it just gets eliminated to a hypothesis. And they say, okay, let me go check all the bacteria or the viral infection that caused that sin. That case is a hypothesis driven. So you have to look at that. I don't care about, any kind of pathogen or bacteria that is in your gut, because the chance of that is just astronomically low. So they go to the hypothesis driven test. And in the case of biosensing, they became the area that I think right now is probably keeping everything on the right track, is a little bit healthy. So, for example, one of the systems that is this system is good, for example, for, it was bioMérieux here in France have this. They bought the BioFire company. So they're looking at, this test was pre-COVID, this existed. This will essentially look at 22 targets for upper respiratory. The multiplex PCR method, so it's an array of, I think, 60 chambers. You run things, different PCR on it, look at the signal and see which one is which. But look at a bunch of viruses, bunch of bacteria and everything. And if any of them is positive, yeah, you will be good. But this test is pretty expensive. It's still $600, by the way. It hasn't gone down. And you might say, why people do $600? Why is this? Why don't we use this test? What is the issue of it? This test, essentially, when they use it, when the patient is immunocompromised. When a 5-year-old has a runny nose, just give them orange juice and just go home to sleep over it. But it is possible. So these arrays were possible. Now, if you look at the case of, let's talk about a little bit of the case of COVID. When COVID happened, we all know, we all experienced it, the craziness and all of those things. And then you had the PCR test. You would go and they just look for COVID. There was nothing else. So it was a single plex panel that they would look, and it was a PCR based. Then this antigen methods came in. You can go to pharmacy, buy them and do the testing. And let's say if the left one is the gold standard, let's run some numbers and see how these tests are different, because it's relevant to the next level of the biochips that if you want to build. There's a huge difference. So let me explain this. What is this graph? If you assume a PCR test just for COVID, it's positive and it's a gold standard. You run that test with different concentration of the virus in the swab, and look at the concentration. 10 to the power of 3 is very low when you have symptoms. This is when you're sneezing, you're a cloud of, sort of floating liquid with the virus over there, right? So actually, these are real numbers. These are from swabs. This is not a fictitious scenario. And these 3 colors are 3 lateral flow tests which are commercially in the market, from BD, Sophia, and Binax, which was from Abbott. And this is a paper that is in a scientific report. You can take a look at it. So what they did is said, okay, let's go and compare this with the PCR. When the high concentration is, they're perfect. When you're sneezing, I can tell you exactly you have COVID. Okay. When the concentration is 70%, when you get 10 to the power of 4, nothing. So when I was in my mom's, sneezing, I said, should I use the BD or should I go to the PCR test? I said, just go do the PCR test. These things are there. So some of these I would call it as a feel-good test. Just remember, it makes you feel good if you want to do it, but really it's not there. So, and these concentrations are the concentrations that you have to actually work. And if you want to beat it. So everybody and their cousin made an antibody test to detect it. Some of them obviously specificity and other performance are better. I don't want to be condescending, but but these are the concentration that you have. So if you look at the problem that I say, this is right now what it is. We know the sequencing, microarrays, and all of that. But I believe if you want to look what is happening and what is relevant is going to be, I doubt that except the fundamental research in our field, we can do things at that scale of parallelism and everything, but probably targeted is the one that I think and has the highest volume right now. And people always have asked me that if we just had this for $60, then I would not do this. Because you would get the answer right away, what's wrong with you. Everybody who has a small kid, when they have infection, you would pay $60. You just want to know that this is not a bacterial or something that you have. But the doctor says, have this antibiotic, which is, by the way, $60. You go and do it, and something happens over there. So I think it's pretty interesting, and I hope we're starting to exponentially forget about COVID. As you walk in the airport, you can see, but hopefully that will happen. So I put actually my bet on it. So it's a little bit, that's the only slide that I plug my own work in it. So I think the previous company and everything, we tried to build on the chip, these type of system. The first one, which was in Silixa, was doing a very high multiplex qPCR, and this was before COVID. We did the upper respiratory, so we were trying to compare that. So it's an array of the fluorescent-based detection and all of them, but you do the amplification on the array, and then you essentially would actually detect it and all of those things. So if you look at the structure of these things, the pixels in each array of, this is a TSMC chip, that you would do, it's all the references that I made. So it's all those components, but you have to make the surface chemistry work, we have to make the emission filter work, you have to make it the thermostable attachment. And so a lot of these works were done at the company, which was the research to make it into a viable or robust biosensor in general. But these are all published work that you can take a look at it. And I'm seeing, and I was trying to get some information about DNA electronics over here. I know they had it, but they haven't published all the detail, but people have tried. And I think it's really not for the weak of heart, because you also have to deal with the regulatory bodies and everything. But in the targeted area, that's probably, a pretty interesting space that you want to go. All right. So I don't know, I'm good. I don't know, I have 2 timers, so okay. Okay, so let's look at the limits. So let's go back to this area. Let's say I'm a CMOS dude and I want to actually decide what it is. This is kind of my trade-off curve. All right, so remember, we have to make these chips, right? And let's say the 2 things that I care about is the pixel size and the array size, right? If the array becomes very large and my pixel becomes large, it becomes bigger than an 8-inch wafer. So this limit of the blue is I cannot have 100 micron pixel and 10 to the power of 8 of them. You cannot manufacture them, right? So this is a hard limit that we have. When you go above some level of pixel size to about, half a micron, you're going to deal with the Poisson noise of molecules binding and everything. You need some statistical averaging to be able to do these things for the array case. So this is a level that is also pretty difficult. Now, if you look at this space, as we talked about it, if your arrays are small, and remember, dashed lines are soft limit. You can do printed probes. If you go higher than that, you have to do random arrays or some other method. And also for the pixel sizes, if you're bigger, you cannot print with a resolution of pitches of less than 100 micron. Mechanically, it's almost impossible to do it. So you have to do in situ synthesis. When you get less than 60 micron, your pixel cannot have that many transistors in it. So you have to put your sensor on an electronic diet. So you have a minimum system in it. When you go more than 10 to the power of 7, you get, you have to worry about the I/O or how you get the information out of it. Here's the punch. The punchline is whole genome sequencing that we talk about it is up there. It's pretty difficult. That's why I put it as great. People have said, okay, probably this is interesting, but not for me. Targeted sequencing is still alive. So you're talking about 10 to the power of 6 pixels or something around that. And if you can get it around, sort of 5 to 10 micron, you're good. But you still have to compete and make a system from start to end. When you get to the DNA microarrays, I don't know if this is an extra, sorry, the type over there. And protein arrays, fine, but nucleic acid amplification testing and things based on PCR or any way that you can look at this precision is probably interesting. And I look at it, and the cost is also, I didn't want to put too many costs and margins in this talk as well. You just have to remember in this space, we're talking about reimbursements of probably $40 to $150. And the person who makes the consumable makes generally, best case makes 1/3 of it. The labs take half of it for the margins, and you have distribution and everything. So whatever, if you want to look at reimbursement of test, whatever number that is, divide that by 1/3, and that's what you can sell it to the lab. FDA approved on all of them. So it's not $150. You will never see all of it over there. And that's different than research market. That's for the diagnostic market that we have. So these are the two spaces that I would stay between that. And the only thing that I would say, I have 2 other slides that I want to talk about it. You know, what my advice, I mean, I don't think I'm in a good position to give the advice, but my take on it, I've seen this field a lot because of funding situation, at least in the US. I don't want to generalize it to Europe and everything. Is limited by the heights, and sometimes we have to relate everything to healthcare and everything. I'm just designing a transistor over here. I have to go and do my research over cancer and outpatient settings in Africa. I mean, what? So sometimes slides like this come out, which is, we laugh about it, but I know it's a necessity, a secret, you know, wink, wink between all the academics, but I think it affects wrongly the students and people on the field, because they say these guys are out of touch, they don't know what is real. But I think, you know, it would be healthy to stick to our guns. If you're designing a good sensor, we're designing the good sensor. And sometimes they have to respond, because the companies have been successful, or at least the academic groups that made an impact were there. They believed in the thing which was correct, and they proved it. Yeah, temporary, maybe short term and everything, because you ended up being a marketing person just making these kind of stuff. And obviously, industry has its own thing. This is from Theranos. I know her personally. So, and you know, if you look at the history of that company, you know, there was a lot of things in that company worked. A lot of good stuff was over there, but they just make something which is, you know, not possible and everything, and they run with it, and a lot of people with money. And when something goes wrong, they just pull down. Theranos, the reason it's personal to me, because we were low cost. So when Theranos went down, the whole industry went down. Nobody wanted to fund it. You use the D word, diagnostic, you're dead. You know, so, and suddenly everybody, the problem that happened to it was the expectation out of the work became irrational and impossible. So you're building a sensor for diagnostic. You're building a PCR system. PCR system, let's say just a thermocycler. You're building a thermocycler. As an engineer, you have to show temperature, uniformity, time, and all of it. But they say, oh, I don't know if this works. You have to show me the clinical result of this work. This graduate student is building a thermocycler. It just, because they got scared of stories like Theranos, that somebody says something, because I don't understand it technically, the only way that I can gauge a value of something to showing that the patients and FDA and all of them says yes. And this is an extremely high bar, because there's so many technical things that you happen. And I think as previous academic and all of them say, the middle matter, you know, we work with numbers. We don't care about patient settings. Sometimes, okay, you know, noise, sensors and everything. And I'm not saying this doesn't affect the end result, but that's the domain that you have to compete with other ones, you know, the other one we have. And they did a lot of damage in these things, but I think I remain hopeful, and I'll end it up with this negative slide, I guess. But, you know, I think, thank you very much for taking it.